students, today's lecture is on cell wall structure of prokaryotes, and we will study mainly on bacterial and archaeal cell wall. So, as we know that bacteria are found in different shapes and sizes, whereas for the archaeal cell structure, lesser facts are known to us. But there are certain features which distinguish both of them. So we are going to study and discuss them one by one. So here we have a typical bacterial cell structure. So this is a typical bacterial cell structure which is having the important labels, but our main focus will be on the cell wall. So this structure is the cell wall, which is the outermost layer of a bacterial cell. Yes, so we have some basic points of bacterial cell wall. Each bacterial cell is enclosed by a rigid cell wall. Yes, each bacterial cell is enclosed by a rigid cell wall. It lies outside the plasma membrane. It is composed of a peptidoglycan layer, and this composition of this peptidoglycan layer varies amongst the bacteria. There are two different types of cell wall, that is, one is gram positive and one is gram negative. And this division is based on the staining procedure which was developed by Christian Gram. Now, gram positive bacteria are stained purple and the gram negative bacteria are stained red or pink. And this staining is done on the basis of absorption of the stain, and it depends upon the thickness of the peptidoglycan layer of the cell wall. So next we come for the peptidoglycan structure. So bacterial cell, as we have studied in the previous slide, is that it is majorly composed of a peptidoglycan layer, which is a mesh of polysaccharide strands. Okay, so here is the chemical structure of peptidoglycan layer. It is a heteropolymer and it consists of the glycan strands which are cross-linked by the peptides. So here we can see that this red and green, this is that structure, a chemical structure, but it is given in a schematic diagram. So these red, green, these are alternative sugar molecules. Okay, that is NAM and NAC. That is, the glycan backbone is composed of N-acetyl glucosamine, NAG, this is the abbreviation, and N-acetyl muramic acid, that is NAM. And these NAM and NAC are joined together by beta 1,4 glycosidic bond. So if this is the NAM, this is the NAC, these are the alternative sugar molecules. So these two are joined by a glycosidic bond which is beta 1,4. Okay, this is beta 1,4 glycosidic bond. Now what is the glycosidic bond? It is a type of covalent linkage that joins a carbohydrate molecule to another group which may or may not be a carbohydrate. In this case, it is a carbohydrate. Now we have the peptide subunits. So there are 4 peptide subunits. Okay, as we can see in the structure, they are alternating in nature, that is L and D amino acids. Okay, so first one is L-alanine, then we have D-glutamic acid, then we have mesodiaminopimelic acid, and the last one we have D-alanine. Okay, so these are alternative forms of L and D amino acids. Now there are certain bacteria which replace this diaminopimelic acid by L-lysine. So we can see that few bacteria may contain mesodiaminopimelic acid as one of the 4 peptides that is present in the peptidoglycan layer, or there may be a replacement by L-lysine. And this a peptidal interbridge is formed to increase the strength and the mesh-like structure. and the cross-link occurs between the carboxyl group of D-alanine and the amino groups of the diamino acids, that is mesodiaminopimelic acid or L-lysine. So from the structure we can see that if this is D-alanine, so if you go for the chemical structure of D-alanine, then the carboxyl group, okay, the carboxyl group of D-alanine will be interbridged with the diamino acids. Which are the diamino acids? There are two diamino acids here, that is mesodiaminopimelic acid and L-lysine. So in case of bacteria that are having diaminopimelic acid, there will be an interbridge between the carboxyl group of D-alanine, and from here there will be the amino group, okay, there will be the amino group, and this there will be a interbridge formed. So this is the interbridge formation. And in case of the bacteria which is having the L-lysine in place of diaminopimelic acid, there will be an interbridge between the carboxyl group of D-alanine and the amino group of the L-lysine, and this is the interbridge structure. So this is the interbridge structure. So next we have the gram-positive cell wall. So as we know that there are two types of bacteria based on the gram staining, that is a gram-positive bacteria and gram-negative bacteria. So first we are going to study about the gram-positive cell wall nature. How does it look? What are the key points that a gram-positive cell wall is having? So here we have a structure of a gram-positive cell wall. The cell wall is thick and it's primarily composed of peptidoglycan layer, which is very thick in nature, that is 20 to 80 nanometer. So here this is a peptidoglycan layer. You can see the schematic diagram. This is the NAG unit, this purple color, and the green color it's having the NAM unit, and this is the peptide bond. Now gram-positive cell wall also contains a large number of teichoic acids. Okay, so these are the teichoic acids which are covalently attached to the peptidoglycan or the membrane lipids. This teichoic acid is the component which gives the cell wall a negative charge. And then there is a periplasmic space which is very smaller in gram-positive bacteria. Now what is a periplasmic space? It is a concentrated gel-like matrix. Now where does it present? It is present in between the outer membrane and the cytoplasmic membrane. So this is the space which is known as the periplasmic space, which is very smaller in gram-positive cell wall. The enzymes produced by gram-positive cell wall are called exoenzymes. Okay. Now next we have the gram-negative cell wall. So this is the structure of a gram-negative cell wall. The peptidoglycan layer is relatively thin in nature, that is 2 to 3 nanometer. So if we distinguish the peptidoglycan layer, we can easily find that the gram-positive cell wall was having relatively very thick layer, that is 20 to 80 nanometers, and comparatively the gram-negative cell wall is having only 2 to 3 nanometer of peptidoglycan layer. And this is the reason that while staining, the purple stain, that is the crystal violet stain or the primary stain, it doesn't get retained in the gram-negative cell wall. And what else does it contain? It consists of an outer layer that lies outside the thin peptidoglycan layer, which is known as the LPS. Okay, so there is again another outer layer which is present outside that of the peptidoglycan layer, and which is called the lipopolysaccharide layer. And this outer layer is rich in a lipoprotein which is named as a Brown's lipoprotein. Okay, so here this is the outer membrane, This is the periplasmic space that is in between the outer membrane and the inner cytoplasmic membrane, and here we have the plasma membrane. So what does this outer membrane consist of? The outer membrane consists of the porine proteins that functions for the permeability of the small molecules like glucose and other monosaccharides. And the LPS layer that we are having, it may serve as an endotoxin and can cause some symptoms that arise in gram-negative bacterial infection. The periplasmic space outside consists nearly of the 40% of the total volume of the cell wall. Now, as we have known that there is again an outer layer which is called an LPS layer, we should know the structure of the LPS layer. Okay, so here are a few points that constitute the LPS layer. The lipopolysaccharides are large molecules which consist of a lipid and a polysaccharide, which gives its name a lipopolysaccharide. So it comprises of 3 parts. Okay, so here as from the structure you can see that there are 3 parts. The first part is the O antigen. This is the O antigen part. The second we have the core oligosaccharide part, and the third we have a lipid A part. So the O antigen or the O polysaccharide, what is it? It is an outer polysaccharide which consists of 25 repeating units of 3 to 5 sugars. So this structure is having like 3 to 5 sugars which have 25 repeating units. These are hydrophilic in nature. The O antigen is highly varied amongst the species. So different bacterial species have different types of O antigens. These antigens are very important, like it is used in identification in some of the gram-negative bacteria, and they are also toxic in nature, and it may account for the virulence, like they contribute to the pathogenesis of the gram-negative bacteria. Then next we have the core oligosaccharide. This core oligosaccharide is of 5 sugars linked through keto-deoxyoctanoate, that is KDO, which is used for the linkage to the lipid A. And the third part we have the lipid A, which consists of 2 glucosamine units. These glucosamine units are attached with 3 other fatty acids and phosphates or pyrophosphates. And these fatty acids, as we can see from the figure, are used to anchor the lipid A to the outer membrane. Okay. Now we have the functions of LPS layer. The outer membrane serves as an impermeable barrier to prevent the escape of important enzymes such as those involved in the cell wall growth from the periplasmic space. It also serves as a barrier of various external chemicals and enzymes that could damage the cell. The outer membrane allows the transport of small molecules such as the nucleotides, oligosaccharides, monosaccharides, peptides and amino acids to pass across via the porine channels. Lipopolysaccharides is a pyrogenic in nature. Now what is the meaning of pyrogenic? That is, which is responsible for fever. As we know that the LPS layer of the gram-negative bacteria is responsible for the pathogenesis also, so it may be pyrogenic in nature and can trigger fever and also causes the endotoxic shock. LPS activates, and how does it do that? LPS activates macrophages, leading to the release of TNF alpha, IL-1, IL-6 and IL-1, which is the major mediator of the fever. Now LPS contributes to the negative charge on the bacterial surface because of its charged sugars and the phosphates of the core polysaccharides. And the lipid A also contributes to the outer membrane stability. So these are the functions of the LPS layer. Okay, the functions of the LPS layer. So next we have the overall function of the cell wall. So as we know that the bacterial cell wall is the outermost structure and it is very essential for the bacterial growth to maintain their stability. It determines the shape of the cell. It protects the cell from the osmotic lysis and the toxic substances and also other pathogens. They help the cells to anchor appendages, means the flagella and the pili or other appendages that the bacteria have. They use it for the locomotion and etc. So these appendages are anchored to the cell wall rather than the cell membrane. So the cell wall also contribute to the pathogenicity, as we have just studied that the gram-negative cell wall, the LPS layer, they contribute to the pathogenicity of the bacteria. And the bacterial cell wall is also a site of action of several antibiotics. Example, penicillin. We know that the mode of action of penicillin is the bacterial cell wall. Okay, so we are going to study about this in details in further classes. So now we come to the archaeal cell wall. Till now we were discussing about the bacterial cell wall, and I have given the basic topics and also from the exam point of view. And now we are going to study about the archaeal cell wall, though we know very less about the archaeal cell wall structures. So archaea are single cell microorganisms that actually lack a cell nucleus and a membrane bound organelles. Like other living organisms, archaea have a semi-rigid cell wall. Okay, so what they have, a semi-rigid cell wall that protects them from the environment and maintains the shape and chemical equilibrium. Okay, the cell wall of the archaea is composed of S layers and they lack a peptidoglycan molecules. So this is the distinguishing features from bacteria, that is the archaeal cell wall. They actually are devoid of peptidoglycan molecules. Their cell wall synthesis is from the S layer proteins. And there are few groups of archaea, that is the methanogen group or the methanobacteria. They have a pseudo peptidoglycan layer in their cell wall, but there are exceptions that they do not have the amino acids and also the N-acetyl muramic acid in their composition. So here we are having a picture. Yes, so here we are having the picture of the archaeal cell wall. This is the cytoplasmic membrane and this is the pseudopeptidoglycan, or we call it a pseudomurine, as they do not have the N-acetyl muramic acid, so we call that they have a pseudomurine. Okay, so this is the overall structure of the archaeal cell wall. So in next class we are going to study about the biosynthesis of the peptidoglycan layer. So thank you for today.